one. Let me grab the other and get ready. So that's the soma or cell body containing all the organelles. The nucleus is this one right here, the big nucleus right here. You can see the nuclear pores in the nucleus. I think nuclear pores is your next thing. Notice that I have a column of functions for this stuff. You have to know those functions too. For example, the nucleus is genetic storage. The nuclear pores is how things get in and out. Ingress and egress of molecules. All right, so that's nuclear pores. Neuroplasm is just the cytoplasm. Neuroplasm is this cytoplasm that you see here. It's just a fancy name for cytoplasm because you're in a neuron. By the way, the cytoplasm in an axon is axoplasm. Now I know it's not on your list. I'm just showing you the trend. The cytoplasm inside an axon is axoplasm. The cytoplasm inside the soma is neuroplasm. It's just simply cytoplasm you learned before. Neisel bodies is rough ER. So you can see the rough ER here. It has the ribosome studded onto it. So this is rough ER. Now let me show you, not all rough ER looks the same. On this model, the rough ER has the red dots on it. The ribosomes are red. And the rough ER has red little dots on it. Okay? So I'll come back to that model in a few minutes. Let's keep going with this model. So Neisel bodies are rough ER. Dendrites. Okay, be very, very careful. This is a dendrite, this is a dendrite, this is a dendrite right here. But what's this turquoise thing? It's not a dendrite.
Telodendron?
Yeah, it's the end of an axon from another cell. The ends of axons bifurcate into collateral branches, and then the collateral branches bifurcate into the ends called telodendria. And actually the very end of a telodendria is called a synaptic knob or axon terminal. This is a telodendria from another axon. This is a telodendria from another axon. It's not even part of this cell. It came from another cell. It's just coming in and it's innervating the cell and telling it to do something.
So it's a synapse then?
Yeah, so this would be a synapse right here. This is a synapse between this synaptic terminal and this soma, absolutely, or this dendrite. So be careful. These are dendrites here. Dendrite, 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 dendrite. Not a dendrite. Not even close. On this model, dendrite, 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 axon terminal. Telodendria, axon terminal. Telodendria, axon terminal from another axon. Is it clear the difference between dendrites that belong to this soma and telodendria that belong to another neuron? Okay, good. I might be getting better at doing that. We'll see next week when I quiz you on this. Okay, the axon hillock. Here's the axon right here. And this dilated region of the axon is called the axon hillock. It's the very beginning of the axon. This is the axon hillock. Now you might wonder why I'm having you learn such a subtle area of the axon called the axon hillock. The reason being is because there's a zone in here called the trigger zone. Now let me explain what that means, the trigger zone. See these telodendria coming into this soma? These telodendria, these other axons coming in and talking to this soma, they're coming in. They could excite this neuron, or they could inhibit this neuron. What if this one's exciting and that one's inhibiting, and this one's exciting and that one's inhibiting, and this one's exciting and that one's inhibiting? What if I have a mix of signals coming in? Some neurons are saying fire, some are saying don't fire. What does this neuron do? Well, it has to add them up. It's called summation. It adds them up, it decides who wins. If there's 10 excitatory signals and 5 inhibitory signals, excitatory wins. The decision is made in the trigger zone of this axon hillock. The axon hillock contains the trigger zone. That's why we have you learn it. Matter of fact, the function of the axon hillock is trigger zone, summation. So this particular neuron is going to sum these signals and either fire or not fire. The decision is made at the axon hillock in what we call the trigger zone. This is the axon. This is the axon. Now look at, I actually had to remove something that I'm going to name for you. Right now, well, actually right now I can see a little piece of the axon. Right now I can see a little piece of the axon right there. But to see more of the axon, I got to take away this fatty sheath, and I'll name that sheath for you in a second. Now I can see the axon better. All right, so that's the axon. All right, the myelin sheath, that's the fatty sheath. This is all myelin sheath right here. All myelin sheath. What happened is this entire, whatever color that is to you, greenish, yellow, yellowish green, whatever color it is to you, this entire thing is a cell. And the cell is called a Schwann cell or a neurolemma. It's called a neurolemmal cell. This Schwann cell wrapped itself around the axon 50 times or so, a ton of times. And in this Schwann cell, this is a different Schwann cell, this Schwann cell wrapped itself around the axon 50 times. When it wrapped itself around the axon, it's like wrapping a tube of toothpaste around a pencil. All the toothpaste gets pushed to the end, right? And the part of the tube of toothpaste wrapped around the pencil is just the container. There's no toothpaste there. Well, when this Schwann cell wraps around the axon, all the cytoplasm gets pushed to the outside. The nucleus of cytoplasm gets pushed to the outside. So what's left in here that's wrapped around the axon is just plasma membrane. What's plasma membrane made of? Phospholipid bilayer. So really what's wrapped around this axon is layers and layers and layers of phospholipid bilayer. Lipid, fat. This is a fatty insulating sheath. This is the myelin sheath right here, which is just plasma membrane of the Schwann cell. Yes, ma'am?
When you quiz us on this, do you want us to say Schwann cell or do you want us to say myelinated?
So if I put a letter here and say name that structure, you would say myelin sheath. If I put a letter here and say name that cell, you would say Schwann cell.
Okay.
So I'll ask you for the cell. You'd say Schwann cell. If I ask you for the structure down in here, you'll say myelin sheath. All right, let me put these back together and show you something. Notice that there is a space between this Schwann cell and that Schwann cell where you can see a little bit of the axon right there. This is a node, and it's called the node of Ranvier. Spelled Ranvier, but it's French, it's Ranvier. This is called the node of Ranvier. It's this little space where the Schwann cell did not myelinate. So this is called the node of Ranvier. All right? What happens is, when the signal goes down the axon, if the axon is myelinated, because not every axon is myelinated, some are unmyelinated. If the axon is myelinated, the conduction of the signal jumps from node to node to node. It's called saltatory conduction. And the depolarization or the signal jumps from here to here, jumps from here to the next node, jumps to the next node. So in myelinated axons, conduction is very, very fast because it doesn't have to depolarize at every little patch of membrane. It jumps from node to node to node. Okay? Saltatory conduction. Telodendria. These are the thin ends of another axon, and then the dilated ends of that axon are called axon terminals. Sometimes they're called synaptic knobs. Sometimes they're called terminal bouton, means end button. So they have lots of names, what I say, synaptic terminals. So this is a telodendria, and that's a synaptic terminal. This is a telodendria, that's a synaptic terminal.
I'm sorry, can you say that again? What's the difference between telodendria?
Yeah, the telodendria are the skinny end, and the synaptic knob is the fat or dilated end. See how the telodendria get fat at the very end? The synaptic knob is the fat part. Synaptic knobs are also called synaptic terminals. They're also called axon terminals. They're also called terminal boutons. Synaptic knobs have lots of names. All right? Any questions about the cell? Okay, before I move on, let me make sure that this cell doesn't have anything funny or weird. No, the Niesel bodies are pretty easy. They have the red dots on them. Pretty easy. Schwann cells with myelin sheaths. Oh, well, that's not till next week. Let me show you something. See this? I'm going to take off the end of this axon. Now, this is the Schwann cell. This gray thing is something else. And I don't believe this until next week. Yeah, it's not. I'm going to tell you anyway. This gray thing is a connective tissue sheath that I'll teach you again next week, which is called endoneurium. Endoneurium. You remember in Bio 203, the connective tissue architecture of a muscle? You had endomysium surrounding a myofiber, and then those myofibers were in a group called fascicles, and you had paramysium around the fascicle, and then all the fascicles were packed into the muscle organ, and you had epimysium around the entire organ. Nerves are going to be the same way. You have endoneurium around an individual axon. All these axons are going to be packed into a fascicle. You have paraneurium around the fascicle, and all the fascicles are packed into a nerve, and you have epineurium around the nerve. It's the same architecture that you learned in a muscle in Bio 203. This is endoneurium. And it's not even on today's list, so let's move on. Next.